In this brief presentation, we're going to have three topics, three short topics that we'll talk about. Glucagon, the last hormone we'll introduce, and then a summary of how all the hormones come together and regulate the different macromolecule metabolism, and then we'll have a brief discussion about appetite. So glucagon first. Glucagon is a protein hormone that is synthesized, stored, and secreted by the alpha cells of the pancreatic islets. It's about 29 amino acids long, and its stimulus for release appears to be a decrease in plasma glucose, although the mechanism isn't as well established as insulin. The inhibitors of release are high blood glucose, insulin will inhibit its release, and also somatostatin will inhibit it. Like insulin, it's transported in solution, has a fairly short half-life of 3 to 4 minutes, and it also binds to fixed membrane receptors. Its effects are to increase plasma glucose by its actions on the liver and adipose tissue, and at high concentrations it will also cause lipolysis. So this graph shows you the different effects that insulin and glucagon have on nutrient levels. So I is to increase, D is to decrease, and so here we have glucose, insulin secretion is increased by glucose increase in the plasma, whereas glucagon secretion is decreased. Amino acids will both increase insulin secretion and glucagon secretion, and that's because glucagon stimulates gluconeogenesis, and so what happens here is that the glucagon turns the amino acids into glucose, and then insulin then stores it as glycogen. So in this case they can actually work synergistically. And then fatty acids in the blood, this will increase insulin secretion but decrease glucagon secretion. And then hormones, gastrin increases insulin and glucagon secretion, whereas secretin only increases insulin secretion and will decrease glucagon. And then cholecystokinin increases both of them, GIP increases both of them, and somatostatin acts as an inhibitor of both. With neurotransmitters, the catecholamines, well, beta adrenergic stimulation increases insulin and but it decreases with alpha adrenergic, whereas adrenergic stimulation of any type will increase glucagon secretion, and therefore glucagon will enhance epinephrine's function, and they go together to ensure that your blood glucose rises when it's needed. Acetylcholine would increase the secretion of both, and stress generally increases insulin secretion, decreases insulin secretion but increases glucagon secretion, again reinforcing epinephrine's role. And then themselves, insulin release will decrease glucagon secretion, but glucagon release is actually stimulatory to insulin secretion for the fact I mentioned earlier on, that glucagon will then convert amino acids into glucose, and then the glucose is then stimulated to be put into glycogen stores with the release of insulin. So this summarizes what glucagon does. It will stimulate the breakdown of glycogen into glucose. It will create glucose from amino acids, and then it will break down free fatty acids and cause the formation of keto acids in the liver. So here, this is a slide that shows the integration of the mechanisms contributing to glucose regulation. And so on this slide you have glycogenolysis, gluconeogenesis, glucose uptake, and glycogenesis, and how the different hormones affect these aspects. So if we look at glycogenolysis, both cortisol and insulin will inhibit that, whereas epinephrine, glucagon, and growth hormone will stimulate it. Gluconeogenesis is inhibited only by insulin, whereas all the others will promote the formation of new glucose by the liver. Glucose uptake by the muscle, adipose, and other cells is promoted by insulin but inhibited by cortisol, epinephrine, and growth hormone. Glucagon will have no effect on this. And then glycogenesis is stimulated by cortisol and insulin, whereas glucagon will actually break it down. And so what that all means is that the occurrence of glycogenolysis and gluconeogenesis increase blood glucose, while glucose uptake and glycogenesis decrease blood glucose. Note that the actions of insulin are generally countered by the actions of the other three hormones. The direction of the change and the magnitude of the change in blood glucose concentration obviously depends on the balance between the competing hormones. So it's not like if you have insulin there that you will get no gluconeogenesis, but having insulin around will reduce it in general. It's the balance of how much hormone is around that regulates what is being created or consumed. Then looking at the effects of hormones on amino acids and free fatty acids, growth hormone and insulin will prevent proteolysis, whereas cortisol will break it down. Epinephrine and glucagon have no effect on this. Whereas with lipolysis, insulin is the only one that inhibits it. All the other hormones will then break down triglycerides into free fatty acids. So regulation of blood glucose has its fundamental purpose to ensure that all cells have the right energy requirements delivered to them based on their metabolic peculiarities. And at the same time, it prevents blood glucose from becoming high enough to cause damage. But the acquisition of calories is under much looser physiologic control, and we're gaining more information about how this happens, but there's still a long way to go. And so, whereas we regulate our energy balance in the body by sort of neuroendocrine controls, how much we eat contributes to that balance, and appetite and satiety are under a complex control of neuroendocrine mechanisms. One of the hormones that is involved in this is leptin, and we can knock out the leptin gene from mice and they lose the feeling of satiety, and as a result they overeat. And so we see here a leptin knockout mouse and then a normal mouse, and you can see that with equal access to food, one drastically overeats over the other. So leptin is a protein, 167 amino acids, that is synthesized by fat cells, and it's produced at a rate that is a function of the mass of the body fat. So the more fat, the greater the rate of leptin release and the higher its concentration in the blood. There are neurons in various parts of the brain, like the hypothalamus, that have leptin receptors, and so therefore receive this signal. And when these neurons are activated, they reduce appetite and therefore reduce feeding and contribute then to this energy balance that the neuroendocrine system is trying to control. It plays an important part in signaling when a developing child is ready to become fertile, and thus is thought to be involved in the onset of puberty. And so here we get a slightly more complicated neuroendocrine control of feeding. Blood glucose sends signals to the hypothalamus, insulin also, and adipose cells, and these are going to then affect your rate of feeding and your energy expenditure. But eating isn't just a homeostatic process. It's psychological, it's cultural, it influences when we eat, what we eat, and how much we eat. Like, we generally wake up and have breakfast probably because we're hungry, but many people get up and they're not really very hungry, but perhaps they'll eat because their family is eating at the table. And same with dinner. There are many factors that lead into when and where and what we eat, and these are clearly outside of neuroendocrine control. But our body does have many signals that add to satiety and hunger, and this graph shows some of them. So we talked about leptin, and that promotes satiety. Insulin will also produce satiety through receptors in the hypothalamus. But then there are also hormones like orexin and ghrelin and ghrelin that will promote feelings of hunger, whereas other hormones, gastric hormones like GLP and cholecystokinin, these will reduce your hunger levels. And so the things that drive your appetite through the day are complicated and involves a symphony of different hormones that both encourage you or discourage you to eating. And all at the same time, we have our cultural and environmental factors that then will determine whether we eat at a certain time or not.